Modes of inheritance of white-eye and ebony-body traits in Drosophila melanogaster, done by Group 4, present it to Dr. Aisha Al-Kayat.
Thomas Hunt Morgan's groundbreaking experiments with Drosophila melanogaster revealed the concept of X-linked inheritance through the discovery of the white-eyed mutation, a trait carried on the X chromosome. This finding marked a significant milestone in genetics, demonstrating how certain traits are linked to sex chromosomes. Understanding Morgan's work is crucial for our project as it provides a foundation for studying the inheritance patterns of white-eyed and ebony-body mutations in fruit flies. The aims of our study: to investigate the inheritance patterns of the cross, identify the traits that are dominant and recessive, define the phenotypic and genotypic characteristics, distinguish the Drosophila mutation, studying how the trait passed from parent to offspring. Now let's discover the life cycle of Drosophila. Egg stage. Fertilized eggs are laid by females and hatching occurs within 24 hours. Larval stage. Larvae go through 3 instars, growth stages, feeding and growing rapidly over 4 to 5 days. Pupal stage. Larvae form a pupa, where metamorphosis takes place over 4 to 6 days. Adult stage. Adults emerge from the pupa, reach sexual maturity within 12 hours, and are capable of reproduction. Cycle duration. The entire life cycle is completed in 10 to 12 days at an optimal temperature of 25 degrees Celsius.
In our experiment, we're crossing a female Drosophila with an ebony body and a male with white eyes, both carrying recessive traits.
Reason of the mutations. The white-eye and ebony-body mutations in Drosophila melanogaster result from alterations in specific genes, critical for pigmentation processes. So how does that happen? The white-eye mutation is caused by a defect in the white W gene on the X chromosome, which encodes a transporter protein responsible for eye pigment precursors. This mutation prevents pigment transport, resulting in white eyes, and follows an X-linked recessive inheritance pattern. The ebony-body mutation is due to a defect in the ebony-E gene on chromosome 3. This gene encodes an enzyme that regulates melanin production. Mutations lead to excessive melanin, causing the body to appear dark or black. It follows an autosomal recessive inheritance pattern.
Materials. For our project, we'll need a brush and clean vials to handle the flies, fly nap to anesthetize them, and a dissection microscope for observation. We'll also prepare labels, Drosophila melanogaster stock, media, and yeast to maintain the flies, and carry out our experiment effectively. Let's prepare the media. We start by adding media containing mold inhibitors like methylparaben, tegacept, and propionic acid. And a blue coloring agent to help observe the larvae is added to the vial, and then we sprinkle 4 to 5 granules of yeast on top, which is appropriate for such larger vessels. Put water, and evenly distribute it. Just like that, our stock is ready. Method. To obtain virgin ebony Drosophila, we start by carefully discarding all adult flies from the vials, leaving only the pupae and larvae. As new flies emerge, we transfer them into empty vials, anesthetize them with fly nap, and examine their gender under a microscope. We isolate the virgin ebony females from this group. Next, we select white-eyed males from the stock, meticulously choosing only those with white eyes. Once we have 3 white males, we place them with 3 virgin ebony females in a fresh vial. Label the vial writing the date and the cross. This group serves as the parental generation, initiating mating to produce the F1 generation. Once pupae begin to appear, release the adult flies from the vial, ensuring only the pupae remain. When the F1 generation starts to emerge, do not wait for a specific number of flies to appear. As soon as the flies emerge, anesthetize them and examine them under the microscope. Record your observations, focusing on their phenotypes, and then discard them promptly to avoid overcrowding or unintended mating. After examining the F1 flies, we carefully select 1 male and 1 female. These 2 are transferred into a new clean stock vial to establish the F2 generation. As with the F1, we allow the pair to mate and produce offspring. Once the F2 flies begin to emerge, we follow the same procedure, observing and recording their phenotypes under the microscope. After completing our observations and ensuring the experiment is properly concluded, we clean the lab and all tools thoroughly to maintain a safe and organized workspace. Finally, we document all our observations and results, detailing the traits observed across generations. And this is our project calendar. The result.
First crosses between white-eyed mutant Drosophila male with ebony-body mutant Drosophila female. The result is all offspring are wild type. The traits align perfectly with our prediction. The Punnett square for first-generation analysis F1 offspring, all Drosophila have red eyes and tan bodies, and the ratio was 1:1. Second crosses between wild-type male and wild-type female from first generation. We predict a 9:3 to 3:1 phenotypic ratio, 9 wild-type, 3 ebony-body, 3 white-eye, and 1 double mutant. The phenotype we get, wild-type ebony-body, white-eye, and double mutant with ebony-body and white-eye. The Punnett square for the second generation, resulting phenotype and ratio, 110 wild-type, 29 white-eye, 35 ebony-body, and 8 double mutant Drosophila.
Let's now observe our data. Chi-square test. Null hypothesis. There is no significant difference between the expected data and observed data of the Mendelian ratio, 9:3 to 3:1. Alternative hypothesis. There is a significant difference between the expected data and observed data of the Mendelian ratio, 9:3 to 3:1. To perform a chi-square analysis, we will use our observed phenotypic ratios. We obtained 110 wild-type, 29 white-eye, 35 ebony, and 8 double mutant Drosophila, for a total of 182 Drosophila. The expected data were calculated, and they are equal to 102.375 for wild-type, 34.125 for white and ebony, and 11.375 double mutant Drosophila. We calculate the chi-square for each phenotype, and we got 0.568 for wild-type, 0.022 for ebony, and 0.769 for white-eye, and 1.001 for double mutant Drosophila. The sum of chi-squares is 2.36, with a degree of freedom of 3 and alpha value of 0.05. The critical value from chi-table is 7.815. Since the critical value from square is greater than calculated value, we are 95% confident that our ratio matches the expected ratio of 9:3 to 3:1.
The first difficulty was during the first generation. A little amount of water we did in the media so didn't grow. We solved this problem by adding some water in the media. The second generation, we added an excess amount of water in the media so the flies stuck on it. We solved this by taking them out of the media by using tweezers with caution to avoid killing them.
Also, we struggled with differentiating between male and female flies. We lost some flies due to incorrect use of FlyNap. After completing the experiment, we realized the value of sharing our knowledge with the community. To make this possible, we designed a website.
Here, we created a very basic website to describe our project and the outcomes we achieved. The project's goal was first stated, and in this section, we included some images along with a brief description of our work and the outcomes we obtained. For instance, what we crossing together, also what we got for the first generation, and what we got for the second generation, and we included what was our double mutant fly we got. Finally, we wrote about the challenges we faced.
We also created social media profiles on Instagram and Twitter to reach a broader audience and engage with people interested in genetics. Additionally, we published a video showcasing the experiment and our results on YouTube, making it accessible to anyone who wants to learn. And that brings us to the end of our project. We hope you enjoyed it and found it informative. As we wrap up, we're grateful for the opportunity to share our journey with you. We hope you gained valuable insights and were inspired by our work. Thank you for following along, and we look forward to sharing more knowledge in the future.